Welcome to the Theories of Aging lecture. I've changed my background. It's probably hot still in the early summer when you might be, or late summer, midsummer when you're watching this. So some beautiful ice-covered trees behind me this time. And so theories of aging, you can read quite a bit more about this in the textbook. I just wanted to cover some basic information. And first, what contributes to aging? Why do we even age in the first place? And we're actually going to talk quite a bit about that in the next couple of lectures. But part of it's just natural. Our cells, our molecules, everything, they grow older. They don't function as well. So we know that that's part of it. We know that there are genetic differences in aging. That some people just genetically age more quickly than somebody else. And so that's based on DNA differences. But your environment can also contribute to the aging process. And think about all of the different things like stress, exercise, diet, exposure to pollution, the kind of life you live, the kind of everything about environment that affects you and your behavior and how you interact with your environment certainly has an impact on aging. I would even include things like access to healthcare in that. And so environment also has an impact and now is going to interact with the genetics. And there's already natural processes going on. And then disease is sometimes part of the aging process, but I haven't read this in a book, this is my own thought, but we've probably all known somebody that disease itself, once somebody got a disease, it dramatically affected the aging process. And so I really believe that there's an independent effect of disease on aging. The theories of aging are broadly categorized into evolutionary theories and then physiological theories. Now, if you think both of these really still fall within biology, evolution, physiology, those are all part of biology. And the evolutionary theories, I've kind of summed up together here. Rather than try and go through them all individually, the actual thought is that we age because evolution never had a reason to fight against it, if that makes sense. Not that evolution does things with a purpose, but through millions of years of evolution, there has been no reason to have organisms live longer. There's not been a need for that. So that's kind of at the crux of all of these evolutionary theories of aging. And you can see it says here, after reproduction, right? I've copied myself. I'm a bacterial cell, I've copied myself. I'm a person, I've copied myself. The parent organism is no longer needed. Now, you might argue against that. That's fine, that's great. We're not here to debate these. These are just theories that have been posited about why we have aging in the first place. But once you've done your job, you've raised your offspring, you've replaced yourself, you can move on and you can die. And so there's never been a reason to keep that process slowed down and have individual organisms live longer. You raised your organism before any deleterious effects happened, so then we can age and die. Physiological theories of aging are a little bit more complex, and you'll see, hopefully you will see as I go through these, that there are interactions even between some of these, that they're not completely independent, isolated theories. And I've taken these from multiple sources and they've categorized them in different ways, and I've categorized them in a separate way that I think makes more sense. The first one, and I'll tell you which ones I like the most, by the way, and one of those is this first one, the telomere theory. I really think there's substantial evidence that this is actually involved with aging and disease and many other things. For those who are not aware, so there are chromosomes that have all the DNA. There are little caps on the end of those chromosomes that kind of keep them from sticking to each other. Otherwise, they'd want to go and bind up next to each other. And these caps prevent that. And those are the telomeres. But every time that chromosome gets copied, so when every time the cell divides, the DNA in the chromosomes gets copied, and the telomere at the end gets a little bit shorter. The next time it gets a little bit shorter, and the next time it gets even a little bit shorter yet. And eventually, that telomere may entirely disappear, or even if it doesn't entirely disappear, they get short enough that it actually starts to interfere with function of some of the genes. And if you're interfering with gene function, as we talked about in the last lecture with proteins, we're talking about affecting different things in the body that can lead to aging. Okay? There's something called the Hayflick principle, and I'm going to bold that here for now. This is something you'll see. If I had vocabulary words, this would be one of the vocabulary words you'd have to define. The Hayflick principle it came up with this guy named Hayflick, obviously, and he was studying cells and found that, cells don't multiply and divide forever. They hit a point where they won't reproduce anymore, or if they do, they're funny. They don't act right. Something's different about them. And they termed this idea cellular senescence. Now, I will give you a clue now. If you're watching this, one of your things to do in one of your reading quizzes is define senescence, but it's talking about a whole body senescence. It's not talking about an individual cell. Here, we're talking about an individual cell. So don't define it exactly the same way as it is here. Cellular senescence is those cells can't multiply and divide anymore and can't do their proper function. And that leads to this Hayflick principle that cells can only multiply a certain number of times. And so if you think about our bodies and our lives and all the cells that we have, some cells are going to multiply more quickly, some not as quickly. And so some cells, some tissues are going to age more quickly than other cells will. The next theory is the accumulated damage theories that I grouped all together. One of the primary things in the accumulated damage comes to free radicals. Okay? If you don't know what free radicals are, they are naturally produced in the body, and they cause some issues. I'm clicking the wrong thing, that's why I won't bold. And we'll talk more about them in detail, so I don't want to do right now, but many of them are produced naturally in the body, and other free radicals are, their pace of production, if you will, is accelerated by external factors. And these free radicals cause damage in the body. We'll talk about that more later on. Radiation. Sometimes we have to be exposed to radiation here or there. For instance, I had my jaw x-rays at the dentist within the last few weeks. They do that, I think, every 2 years. But they're doing that because we want to make sure that my teeth are healthy. And so any other risks are, I think, acceptable with that. But radiation can cause damage over a period of time. And some radiation we're constantly exposed to. There's just radiation that comes from different sources. And people have even been worried about cell phones and the radiation that they produce. Any electronic produces radiation. Toxic chemicals that we can be exposed to more or less. Some we're ingesting even with our food. Some we will breathe in. Some might be exposed to in other ways. But these can cause damage over time. Again, we're talking about accumulated damage. It doesn't always have to be things that come in and cause problems, but what about accumulated DNA errors? I can't remember the numbers, but it's something like 1 in 10 billion. When we copy DNA, we only get an error once in 10 billion. But over millions of DNA and over millions of DNA of cell replication, some are going to creep in, and that can lead to proteins that are dysfunctional. So that's damage that occurs over time, because once you've created that error, if it's not fixed, when the cell copies again, it maintains that error as well as any additional one. So over time, they can build up. There's a waste product called lipofuscin that primarily concentrates in the heart and in the brain, of course, our two most critical organs. And it concentrates inside the cells and begins to interfere with cell function. There's a protein product called amyloid that can accumulate between the cells. And this is actually what happens in Alzheimer's disease. It's accumulating between two neurons, and those now have a hard time communicating with each other. And they find these amyloid plaques then in Alzheimer's disease. Calcium. Calcium is in every cell of our body. It's an abundant mineral, the most abundant in the body. And over time, that calcium can begin to accumulate in cells. And in fact, that's a natural part of the aging process that leads to a term you've probably heard before, arteriosclerosis. There's also something called atherosclerosis, which is a type of arteriosclerosis. But in general, arteriosclerosis is hardening of the arteries. Why are they hardening? Well, because calcium is getting deposited in there. And over a long period of time, that can cause some issues. All right, so I mentioned at the top here that free radicals is the telomere theory. I place a lot of emphasis on that as a theory that I think is responsible for a lot of aging. I think the free radical damage is highly responsible. And then I'm going to come down to something called mitochondrial damage. And I mentioned this one, and we'll actually come back and mention this because this one actually connects with some of these other theories. Mitochondria is actually the source of most natural free radical production. It happens in the mitochondria. And the way I describe it, you might even hear me say this again, is we produce free radicals because we eat and we breathe. We burn the energy, we produce ATP, that's happening in the mitochondria, that's where those free radicals are produced. And because of that, mitochondria are also probably the most damaged by free radicals because these free radicals don't float all over and start to do damage everywhere. They can, but they're going to do their damage closest to where they are, and that's the mitochondria. Well, what do the mitochondria do? They produce your ATP. That's critical. In addition, mitochondria are easily affected by radiation, pollutants, medication, and so they're just damaged more easily. And so over a long period of time, from birth to, whatever age, those damaged mitochondria can accumulate. And sometimes we replace them, sometimes we don't. And we'll talk about this more again when we talk about our next topic. But this links with free radicals, and it can also link with telomeres. And one of the reading articles that you do a reading quiz on will actually mention that and tell about that relationship. The final physiological theory is called the rate of so just to recap, we have the telomere theory, the accumulated damage theory, and now with the rate of living theory. And the accumulated damage had multiple parts there. But the rate of living theory states that if there's a higher rate of metabolism, that increases the aging rate. Well, if we know that metabolism is linked to free radicals, that might actually make some sense there. And so therefore decreasing longevity or the maximum amount of time that somebody can live. Well, the way they measure this is based on oxygen consumption because oxygen consumption is actually linked to metabolic rate. The higher the metabolic rate, the more the oxygen consumption. Now, I don't mean that this is true across humans, but where they have shown this is across species. Although it's not 100% accurate, they've shown some pretty good correlations where the animals with the highest metabolism actually have the shortest lifespan. Those with the lowest metabolism have the longest lifespan. Interestingly, humans actually fall off the line for this. They put all other animals on there, from like a mouse to an elephant. The mouse has a lifespan of 3 years. The elephant has a lifespan of 80 years. And they can put all the species in between on there, and it pretty much well matches up. And I'm doing it based on size, right? But that size is also related to metabolism. Right, the rest of this is just some basic information here. Homeostasis. This is a key term for biology. Where we keep, this is the way it's described, a body's internal conditions remain relatively constant. Blood glucose levels aren't going to have these wide ranges. The amount of oxygen in your blood is not going to have this wide, massive range. We don't go without food for 2 weeks and then binge. No, we're really pretty consistent with the amount of food that we eat to provide the energy that the body needs. We maintain a relatively constant body temperature. Uh, just all of these things are controlled, and is primarily through negative feedback systems, where you detect a change and the body then reverses that change. That's a negative feedback system. That's about as simple as an explanation as you can get. The problem is, as we get older, this is true. We cannot maintain homeostasis as well. But some organs will be more affected than others because of this idea of reserve capacity, right? If I only need 50% of my organ function in order to maintain homeostasis, well, I can actually lose 50% of my function before I actually see an effect. That extra 50% is the reserve capacity. And so there's no effects of aging until that reserve capacity has been exhausted. And at that point, it can actually affect things like quality of life. I always like to say this class goes beyond the biology because it's applicable. Quality of life is not a biological measure. That's a social, psychological, clinical measure, but it relates to the biology. If we can't maintain homeostasis, we don't have as good of quality of life as we could otherwise. And every organ, of course, could age slightly differently. Senescence, you'll find a lot of different definitions for. We gave one for cellular senescence earlier, but this is like whole organism senescence. I defined it as age changes that cause detrimental effects. It's not senescence. Just because somebody's aging doesn't mean it's senescence. It's when it begins to be detrimental that we can determine as senescence. Biological versus chronological age. I'm sure we've all known people that aged quickly. I went to a high school reunion, I guess 3 years ago. I was fortunate enough to win the Fountain of Youth Award for still looking younger than I was, that I hadn't changed nearly as much. And I was age 50 at this time. But there were some of my classmates that looked way older, 65 they looked, like they'd had really hard lives. And maybe they had, or maybe it's just that they aged more quickly. Everyone ages differently. Some are slower, some are faster. Remember, we had genetics, we had interactions with the environment, and that includes lifestyle. So all of those things play a role in aging. Organs age at different rates, right? Some organs have a huge amount of reserve capacity, and others don't have so much. And so you might see some differences in how they function with age. And remember, we don't see any changes until the reserve capacity is gone. Well, biological age is really how old is that body compared to a normal person? What has changed about them? How much reserve capacity is left? Whereas chronological age, hey, as I'm recording this, I'm 53. That's not going to change. Am I younger biologically? I'd like to think so, but we're all different. And that's the difference between biological versus chronological age. And that's kind of important to keep in mind. As we talk about everything in the class, we'll talk about, well, this happens at this age, and this happens at that age, and that happens. Well, not for everyone. Some people it might actually happen earlier, some it might happen later. In some people, it doesn't even happen at all. Right, mean longevity versus maximum longevity. It is believed that the longest humans can live, which would be maximum longevity, is about 130 years. That we really can't live longer than that all natural processes will lead to a 130-year death. Mean longevity is really the average of how much we actually do live. And that's, of course, different by sex, it's different by race, it's different by socioeconomic status, it's different by country. It differs based on a lot of things. It differs based on lifestyle habits. In studying aging, and many students that I've run into have already been familiar with these kinds of studies, especially if they're coming from outside biology. But a cross-sectional study is where you take individuals of different age groups and you compare them. right? That's easy to do because we can grab the people, we can study them, but we're not studying the same people, so we can see differences, but they're not as strong. Longitudinal studies follows the same population over time, and that way we can actually see how people change over time. But you can already see that's a tough study. If you want to study them for 5, 10, 20 years, you've got to have the funding, you've got to keep them in the study. Not really easy. It's a much better study, but much tougher to do. So we don't see too many of these. A cross-sequential study actually combines those two, and there's a lot of major studies that actually do this that are fairly well funded, where you take all kinds of groups of people and you follow all of them over time. So now you have different ages and you're studying all of them over time, and that gives so much more information. But you'll actually see in this class that we learn a lot from animal studies. Are animals people? No. But we believe that the basic mechanisms of aging are true across species. And Drosophila, because they have about a 2-week lifespan, Drosophila are fruit flies. They're pretty easy to study. Yeast, actually, and that's not an animal, but yeast is actually closer to humans than plants, and actually exhibits some of the same aging characteristics. Mice are often studied. Rats are studied. As you get closer, macaques and chimpanzees are studied. And so we learn a lot of different things from these animal studies, because a mouse lives 3 years maximum. You can do so many more things and study it. You just couldn't do that in a human. And that's the end of this particular lecture.